Yes, so good evening. We will go through test 6 discussion. So test 6 is mostly covering physical anthropology. That too, first portion, physical anthro, we are covering 1.4, mostly 1.4 Darwinian theories, and then 9.1 to 9.4. This is the scope of this test. So questions are quite easy and repetitive. Some of the questions are, there are two kinds of questions. Some are heavily anthropology based questions. Some are more biotech, S&T, biotechnology based questions. So these questions I'll give you an outline and quickly close it. But these are the questions where we can have more discussion, because these questions, they can come, but the chances are less. Questions like recombinant DNA technology, or questions like DNA fingerprinting, or CRISPR-Cas9. These questions we will discuss. We will not discuss it great length. I'll give you an outline, right? First, so we'll begin with the first question. Pre-Darwinian theories on evolution. I was surprised most students had difficulty in writing good answer for this. This is a very straightforward question. Just that students don't read pre-Darwinian theories well. The only pre-Darwinian theories we read is Lamarckism. And beyond that, we don't take things seriously. That's why I think many students have difficulty. So when we say pre-Darwinian theories, you have to touch at least some of these core theories. So one of the first person who contributed more to pre-Darwinian theory is James Hutton. And then Charles Lyell, Robert Hooke, you can skip, George Cuvier. So these are called catastrophism, uniformitarianism, creationism. These are the theories. So James Hutton's theory, Charles Lyell's theory is also called as uniformitarianism, and then George Cuvier, his theory is called catastrophism, and then Carlos Linnaeus's classification. Linnaeus is the first to provide a taxonomy for plants and animals, and then you can add Erasmus Darwin. He is the uncle of Charles Darwin. Erasmus Darwin's theory, and then you can add Lamarck. So all these theories should come. So who are the people? 1, 2, 3, 4, 5. These 6 people should come in pre-Darwinian theory. And of course, in this discussion, generally we give a lot of importance to Lamarck and his ideas of theory of inherited passing of inherited characteristics. So we can discuss all these theories. So all these 6 must come in the answer. Next question is adaptive radiation. First, define adaptive radiation clearly. What is adaptive radiation? It's very simple. First, give definition. It is a mechanism of, what is the definition? It's a mechanism of macroevolution. It's a mechanism of macroevolution where a single ancestral species radiates into multiple species. Mechanism of macroevolution where a single ancestral species radiates into multiple species. That is what we mean by adaptive radiation. And then give examples. There are so many examples of adaptive radiation, and the most famous example is Darwin's finches, or the finches on Galapagos Islands. Or finches on Galapagos Islands. So here you had initially one set of finches. Then you had different radiations. Some were insect eating, some were seed eaters, and some became cactus and plant eaters, and so on. So this is how they radiated. Another example is of course primates. You can give another example, that is primates. How primates evolved into prosimians, New World monkeys, and then Old World primates, Old World monkeys, then apes, and then humans, so on. So this is the examples. Then you have to discuss the conditions. So very simple. Definition, then examples of adaptive radiation, and then discuss the conditions for adaptive radiation. What are the conditions that are needed for adaptive radiation? Finally, essentially adaptive radiation is triggered by emergence of ecological opportunities. Triggered by an ecological opportunity. It is triggered by an ecological opportunity or emergence of a new adaptive zone. This includes three things. What is this ecological opportunity? And the three things that it involves. First, a new habitat should have opened up. A new habitat should have opened up, and this new habitat not just opening up, it is relatively isolated because there should be no other species existing in that habitat. If there is a pre-existing species, then there is no space for a new one. It should be relatively isolated, and the new habitat should also have a wide availability of niches. It should have a wide availability of niches, so that an animal can occupy different niches. Like one can be seed eater. Niche, for what is a niche? For example, this is a different niche. One can be seed eater, another can be insect eater, another can be plant and cactus eater. These are the different niches. So the different niches should be available. These are the conditions for adaptive radiation. In conclusion, you can write adaptive radiation leads to speciation in the long term. That's it. So connected to speciation, macroevolution and all these terms. Next is pedigree analysis. This is a very simple concept. So pedigree analysis, first introduced by saying that it is a construction of family tree that studies the transmission of traits from one generation to another. Basically, we are trying to explain, what are we trying to do? We are trying to explain the mode of inheritance. And how are we doing it? What is the basis for pedigree analysis? The basis is Mendelian genetics. So we are trying to explain the mode of inheritance based on Mendelian genetics. That is the essential idea of pedigree analysis, and the whole discussion starts with something called as a proband. A proband is a person from whose reference we are studying, and then describe briefly the different symbols, male, female. You need not write all the symbols, but at least the essential symbols you can show. What are the essential symbols that you are showing, and how do you show an affected person? How do you show a carrier? Show these two. And then discuss some of the, at least two types. At least discuss first, list out the different pedigrees that we have. How many pedigrees we have? Broadly, we can have 6 pedigrees. Autosomal, dominant, recessive, and then we have, yes, X-linked, again X-linked recessive, X-linked dominant, and then we can have Y-linked. Lastly, we can even have mitochondrial inheritance. that is inheritance based on mitochondrial DNA. So these are 6 possible types, and then take any 2 and discuss them and write them briefly, at least one. You can take autosomal dominant, is very easy, draw the pedigree and then list out the broad symptoms, or you can take autosomal recessive and list out what are the broad symptoms. So that's how we can take pedigree analysis, and then also explain, don't just explain this, also you can express the drawbacks. What are the drawbacks of pedigree analysis? What are the drawbacks? Because no method is perfect. One, time taking. One is clearly time. Second drawback for pedigree analysis is the privacy or confidentiality of families. Second, pedigree analysis is not conclusive. We don't get a conclusive idea of which disease the person has. You may say autosomal dominant, autosomal recessive, but in autosomal dominant there are so many diseases. How do you know which disease is going to be there in the family? Or it will say X-linked dominant or X-linked recessive, but within X-linked which disease specifically? So you can't have precision. And other concerns can be issues like disputed paternity. So these are some of the concerns with autosomal pedigree analysis. That's it. Then genetic imprinting. This is a very simple question. I'll quickly discuss the framework for genetic imprinting. This is 1E. So begin by quoting Mendel's theory of equivalence. Mendel's theory of equivalence is that even in case of reciprocal cross, according to Mendel, the outcome would be same. What is reciprocal cross? Reciprocal cross means whether you inherit a trait from mother, whether you're inheriting a trait from mother or father should not make a difference in terms of how the trait is being expressed. That is reciprocal cross. So you get a gene from mother or the gene comes from father, the gene's expression should not be based on who the parent was, male or female. That is reciprocal cross. Let's say you have Huntington disease. The gene can come from mother or father, but there should not be a difference in terms of how the gene gets expressed, whether based on mother and father. Let's say the gene comes from father and it is more severe, but when it comes from mother it is not so severe. Then that is what we call as genetic imprinting. So genetic imprinting means the gene's ancestry should not have a bearing on its expression. And theory of equivalence of Mendel says that the sex of the parent is irrelevant. The gene is the only one that matters. But genetic imprinting shows that sometimes sex of the parent is also relevant to the gene's expression. So what we see is that in some cases, allele from both parents should have equal chance of expression. That is theory of equivalence. Now we should explain why this is so. How is it that whether a gene comes from mother or father, how does it have a bearing on the expression? So we have to explain this. What is the mechanism? The mechanism has to do with something called as DNA methylation. So when DNA is being passed from parent to offspring, the DNA is methylated. It is covered by methyl groups at both the ends. This methylation happens to protect DNA. The purpose of DNA methylation is to protect the DNA. And what we know is that maternal genes are more methylated. Maternal genes happen to be more methylated. Because of more methylation, maternal genes have late expression and low intensity of expression. So because of more methylation, it has late expression and low intensity of expression. Late expression and low intensity of expression. And then give some examples of genetic imprinting. What are the, so first define it, in definition talk about theory of equivalence, then discuss the mechanism, and then give some examples. So what are the examples? First is Huntington disease. It's a clear case of genetic imprinting. In Huntington disease, if the gene comes from paternal, what we see is that there is more intensity. The gene is coming from father, we see that there is more intensity in the expression of the trait than if the gene comes from mother. That is what we mean by Huntington disease's genetic imprinting. Similarly, there are two other diseases. They are slightly named differently, but they are similar. Angelman syndrome and Prader-Willi syndrome. Prader-Willi syndrome, genes come from father, it is paternal, and that is why both have similar symptoms. In case of Angelman syndrome, it comes from maternal side. So in case of Prader-Willi syndrome, it is paternal, and as a result here, those who suffer from Prader-Willi syndrome have more severe symptoms than those who suffer from Angelman syndrome. That is genetic imprinting. And then discuss broad features. What are the features of genetic imprinting? First, imprinted genes expressed differently. Then genetic imprinting GI is erasable. Let's say you got a gene from your mother, which means on the gene mother is 6 is stamped. But then when you are passing the same gene to your children, let's say you are a male, your mother's 6 gets erased and your 6 gets stamped. That is what we mean by genetic imprinting being erasable. So offsprings stamp their own sex. That is another point. Second, it is not a rule. It is seen in only some genes, for some genes only. It is not like mandatory for all. Next, it is also species specific. That is, which genes are imprinted depends on the species to species. Genes which are imprinted in humans may not be the genes that are imprinted in other animals also, and it is seen among higher animals and flowering plants. Mostly, it is seen mostly in higher animals and flowering plants. So that is how you can handle genetic imprinting. Next, we move on to cell division. Cell division, again, not a very important question, but I think most of you can handle this. Not very difficult. Just describe cell division is of two types, mitosis and meiosis. You can, for these kind of questions, cell division, protein synthesis, just have the outline, because this is not really anthropology, it is mostly basic biology. So discuss the mechanism and then talk about mitosis, draw this diagram and show what happens in mitosis. You begin with a single mother cell and end up with two daughter cells. So you begin with diploid number and you end up with diploid number. So identify these two things and show the division. But meiosis is where you have to give more importance, because meiosis is different. And say meiosis, this happens in somatic cells. Meiosis is largely dealing with gametic cells. This is about growth. This is about reproduction. So in meiosis, trace the two steps. Say there is meiosis 1 and there is meiosis 2. in meiosis 1, you have separation of the two chromosomes which are in the same pair, and then in the next step we are separating the sister chromatids. So that is the difference between first cell division and second cell division, and as a result you are ending up with 4 daughter cells, but each not having diploid number but each having haploid number. So, and then the most important point that I think all students missed is crossing over. Nobody talked about crossing over. How the importance of meiosis is crossing over, which happens like this. Because of crossing over, there is genetic recombination, that is, two chromosomes, when they cross over, there can be exchange of genetic material between them. And it is because of crossing over that though children resemble parents, they are not exactly identical to parents, because each child has its own unique set of genes as a result of genetic recombination. So recombination or crossing over must be highlighted. You can't mix it. So this is how, when you have crossover, there is exchange of genes between two chromosomes. So let's say in one chromosome you have AB, other small a, small b. After genetic recombination, these two are exchanged in this fashion. So crossing over leads to genetic recombination. We have to highlight that aspect. And then the next question is twin method. Twin method is very easy. I'll again give you the framework. So in twin method, first talk about the meaning and types of twins. First of all, talk about the purpose. Why are we using twin method? We are using twin method to study the relative influence of gene and environment for a given trait. The whole purpose of twin method is to study the influence of role of genes and environment in a given trait. So talk about the meaning and the different types of twins. Here we have monozygotic twins and we also have dizygotic twins. Explain what these are. These are also called as identical twins. These are called as fraternal twins. So identical twins and fraternal twins. And then we have to discuss how do we determine zygosity. How do we go about determining zygosity? There are broadly 4 methods. First is called placental method. In case of placental method, what we do is that there are 3 layers, but if the twins share single chorion, then there is 70% chance that they are monozygotic twins. That is called as placental method. It is based on chorion. We can't diagnose by amniotic layer, so we generally go by chorion. Second is called similarity method. Similarity method is based on things like identifying whether they have same blood group, identifying their HLA makeup, identifying their serum proteins. If they match, then it's a sign that they have the same zygosity, or based on things like taking skin grafts and seeing whether the skin grafts will match with each other. That is called a similarity method. Third method is statistical method. Here you have a predetermined statistical indicator of how many children in a given population are likely to be monozygotic, how many are likely to be dizygotic, and based on that indicator you calculate this. And fourth is based on advanced genetic methods like DNA fingerprinting. That's how we do this. So first define it, purpose and then meaning of the twins, then determining zygosity. After that, we talk about heritability estimate. So finally, using this twin method, we calculate what is called as a heritability estimate for each trait. So we have to identify what's the heritability estimate for each trait. So heritability estimate is nothing but trying to tell us the proportion of the trait that is influenced by gene and environment. That is called as heritability trait. And to calculate this, we do two things. First, we take monozygotic twins who are raised in different households. Monozygotic twins who are raised in different households. And second, we take dizygotic twins who are raised in same household. This will give us the role of, because they are in different households but same genetic makeup, this gives us the role of environment. This gives us same environment. Here environment is fixed, so here we will get the role of gene. So that's how we calculate heritability estimates. And in the conclusion, you can say that pedigree analysis is used for multifactorial traits. Unlike single factor Mendelian traits, pedigree analysis is used to study the so-called multifactorial traits, where the role of both these multifactorial traits are polygenic traits, multifactorial, or you can also call them polygenic traits. These are governed by both gene plus environment. Examples we'll discuss again in polygenic inheritance, but examples can be things like skin color, hair color, intelligence, diabetes, all this. So that's how you can talk about twin method. Next is DNA fingerprinting. Again, very easy question, but this is, like I said, it is more of an SNT question. So I'll give you the basic outline without going into the details. First, for any of such question, give the right intro. What is DNA fingerprinting? In intro, you can simply just say who developed it. It was developed by Alec Jeffreys, a UK scientist. In India, it was made popular by Lalji Singh. Dr. Lalji Singh is called as the father of DNA fingerprinting in India. And then give principle. What is the principle behind DNA fingerprinting? The principle is quite simple. The principle is that in everybody you have something called as introns and exons. So when you take any person's DNA sequences consists of introns, exons. Exons do not have any coding, any useful material. It is the introns that have lot of useful genetic code. But then exons are useful in the sense that they have a repetitive sequence, repetitive protein sequence like ACG or GAC. There is some sequence that is repeating. So it is based on the principle that though there are introns and exons, an individual inherits most of his genetic code from either of the parents, but the individual also has some unique sequences. These unique sequences are called as STRs or VNTRs. STR is standard tandem repeats. VNTR is variable number tandem repeats. So STR or VNTR. you'll have this unique sequence that repeats in any person's DNA, and this sequence is unique to that individual. So you can calculate what is the sequence that is repeating and how many number of times is the sequence repeating. Based on that, we do DNA fingerprinting. So what is the principle? Individual has introns, exons. Exons have some repetitive DNA sequences. These repetitive sequences, though an individual inherits 99.9%, 99.1% of his DNA from both the parents, that 0.9% is unique to the individual, and hence the sequence tends to be unique to each individual. This repetitive sequence tends to be unique to each individual, and based on this sequence, we are trying to establish that person's identity. That is the principle. So, talk about intron, exon, and then unique sequences, especially these two words must be there in the answer, VNTR and STR. And then only identical twins also share exactly same STR or VNT. Except for identical twins, nobody else, even siblings or offsprings and parents, do not have the exactly same STR or VNTR. And then discuss the steps in DNA fingerprinting. What are the steps? First, you have to do DNA extraction. Second, you use restriction enzymes to digest DNA and break them into smaller fragments. Once you broke them into smaller fragments, we can use gel electrophoresis to separate the DNA into based on size. So you arrange the DNA in a line based on their size using this so-called gel electrophoresis, and once you arrange them, we take an impression, or this impression taking is called as Southern blotting. Then we have Southern blotting, where we take the impression on a nylon membrane, and once we take this blotting, then we do a hybridization with a radioactive probe. Hybridization with the radioactive probe. And 6, we do an X-ray to identify areas of reaction. That is to see where this probe has actually reacted with the DNA, and that impression gives you an exact DNA fingerprint. That is the unique, because this hybridization, that is probes are ready-made sequences. So the way in which the probe binds or reacts with the given DNA varies from one individual to another individual. If person A has, let's say, ACG, person B has CGA instead sequence in this STR. So the binding of these radioactive probes will be different for each individual, and based on how that probe has bound with these particular sequences, we get a unique DNA fingerprint that we then use to establish the person's identity. And then discuss the, after identifying the method, discuss the applications. Applications are there are various applications, starting with forensics, and then we also have we are using this in paternity diagnosis, we are also using this in wildlife conservation, we are using this in immigration, so on. So numerous applications for DNA fingerprinting. So we can use different applications like forensics, parental disputes, missing persons, immigration to a certain identity, and also it is used in forensic wildlife, that is identification of dead animals, poached animals, or in identification of endangered species. So that's it. So what is DNA fingerprinting? Who found it? What is the principle? What is the method? What are the applications? That's all. Next question is genetic load. This is very easy, so I won't bother to explain this. I'll just give the framework. First, what is the meaning of genetic load? It is the extent to which a population's fitness has been reduced from an ideal state. That is genetic load. Next, source. What is the source of genetic load? The sources are again the same factors: mutation, migration, all of these. And once these create the load, natural selection acts on this load and distributes them. Now we have to discuss types of genetic load. Here you have different types: mutation load, migration load, incompatibility load, segregation load, and 5, inbreeding load. Then after types, discuss how it is measured. Measurement: genetic load is equal to 1 minus omega bar, where omega bar is the mean fitness. Omega bar is the mean fitness. And then significance. Discuss significance of genetic load. Is load really good or bad? So in significance, first, if you have very high genetic load, it is danger, because very high genetic load means immediate survival is at risk. People won't survive. If you have very low genetic load, that is also a problem, because if you have very low genetic load, it means there is very less genetic diversity in the individual. Third, that's why if you have a medium genetic load, it accomplishes both. A medium genetic load leads to a fitter population in the long run, not immediately. How? There is some reserve of genes which are dangerous, but these genes are dangerous today, but in the future, if the environment changes, these same genes can become beneficial. But if you have very high genetic load, then it means high number of defective genes, which means the population will not have future survival. But if you have very low, then there is very low diversity. So having a medium genetic load is healthy. So conclude by saying that genetic load is good for the population, though it may be bad for the individual. For the individual it may be bad, but for the entire population it is good. That's how we can conclude this answer. Now Down syndrome, Edwards syndrome is very simple. I will discuss that with the other question 5a, where we also have another question on chromosomal aberration. So I won't discuss this right now. We will discuss 2e, Down syndrome and Edwards syndrome, with 5a. Next, 3a. 3a is Hardy-Weinberg equilibrium. Discuss the factors that produce and redistribute variation. Very simple classic question. So first, I think somewhere we missed protein synthesis, isn't it? We didn't discuss 1D, I forgot. So this is how we can answer protein synthesis 1D. We will first quickly discuss protein synthesis and go to this. Again, these 1.7 questions, don't try to get into too much technicality. Just write with minimum number of facts where you are but getting a sense of completion. Questions like protein synthesis, cell division, DNA replication, all of these you have to answer with a particular framework. Don't try to complicate it. First, define it. What is it? It is a process of creating new proteins. Next, we have three major steps in this process of creating new proteins, and within this we have three major steps. What are these three major steps? One is transcription. Second is translation. Third is post-translation events. What happens in transcription? In transcription, nothing but an mRNA reads the code from a DNA. mRNA reads the code from nuclear DNA and brings it out of the cell into cytoplasm. So transcription is happening within nucleus. This process is happening inside nucleus. And mRNA brings that message out into cytoplasm, and translation happens in cytoplasm at ribosomes. At ribosomes, the mRNA messenger RNA brings a message, and that is translated by what is called as tRNA or transfer RNA. So you must highlight the role of mRNA as well as tRNA. And tRNA, by decoding the message of mRNA, creates amino acids. It creates amino acids, and one after another an amino acid chain is eventually created. One after another an amino acid chain is created like this. So this is a classic big figure. This here, this long segment is the mRNA, which includes the entire message from nuclear DNA. Then this is ribosome, where the whole translation is happening, and transfer RNA translates the message from this mRNA, and based on the message here, it creates an amino acid. Next, another transfer acid creates another amino acid. In the end, ultimately what's happening is that 1, 2, 3 and so on. So these individual amino acid molecules are all eventually tied together to create an amino acid chain. And when the amino acid chain becomes significantly long, you get a protein chain. That's how protein synthesis happens. That's it. So don't complicate it. If possible, just define it and discuss the three major steps, location where they happen, and the molecule involved in each step. It is mRNA in transcription and it is tRNA in translation. First step happens in nucleus, second step happens in the ribosome, and the last step is to join them. And the third step is called as post-translation events. Here, sometimes protein folds occur. Protein folds occur to improve the protein's functionality. That's it. That is all we need to know about protein synthesis. Now we move on to 3A, Hardy-Weinberg equilibrium. So very simple. I think we all know Hardy-Weinberg too well, just that we need to find the right structure for this. That's the only problem. So what is the structure for Hardy-Weinberg? Introduction. Hardy-Weinberg is a mathematical model. So in introduction, use words like mathematical model. In which area? This is used in what is called as population genetics. So it's a mathematical model in population genetics to understand the direction of evolution. Simple. And then Hardy-Weinberg then discuss the 5 conditions. Hardy-Weinberg, and also use the word, it's a hypothetical condition because it's not a real condition. It doesn't exist in the real world. It's a hypothetical scenario. And then there are 5 conditions for it to hold true. Discuss those 5 conditions. What are those 5 conditions? These are the 5 conditions. 1, we should have an infinitely large population. 2nd, no mutation. 3rd, no gene flow. 4th, natural selection is not operational. 5, we are assuming that the mating is going to be non-random. So these are the 5 assumptions that Hardy-Weinberg make, and if that assumptions hold true, then they believe that if these assumptions exist, if these conditions meet, then they propose that the allelic frequencies won't change from one generation to next. That is, they say that there won't be any evolution as long as these conditions prevail. But this is, like I said, a hypothetical model, but in reality, no population has, in general, doesn't have any of these conditions met. So in real world, populations rarely exist in equilibrium. The only case that we know to exist in some kind of equilibrium is PTC testing. That's it. And after this, give the Hardy-Weinberg equation. That is, they begin with a simple trait. They begin with a simple assumption that P plus Q is equal to 1, where P and Q are allelic frequencies. They begin with allelic frequencies for a trait which has just true alleles and follows Mendelian equation. Now, P plus Q is equal to 1. When you go to second generation, then the equation becomes this, because 2P and 2P's and 2Q are involved. And when you open this up, it becomes, until here it is straightforward. So Hardy-Weinberg says, in case of genetic equilibrium, when the genetic equilibrium is holding true, then these terms are nothing but these are the phenotypic ratios. So Hardy-Weinberg are saying that we begin with allelic frequency and we are ending up with phenotypic frequencies. That is the insight. So this step, from where we are assuming that P square is AA, 2PQ is capital A small a, Q square is again small AA. This assumption can be made only in the conditions of genetic equilibrium, when the allelic frequencies remain unchanging from one generation to another. Once we know this, the rest is easy. So you can show the same thing is shown here, and here you have the entire derivative. Take any, if you have the time, to this 20 marker, if there is time, so you can briefly take any one trait and show how, starting with phenotypic frequencies. Generally we begin with phenotypic frequency, and the idea is to calculate allelic frequency. So let's say there is a trait like, what is the trait they have chosen here? PTC testing. Let's say there is PTC testing. You can actually use this. So we know that 36 individuals are PTC testers. It's a recessive trait. So AA is 0.36. If AA is 0.36, this we know based on population studies. If AA is 0.36, that means Q square is 0.6. If Q, that means Q is 0.6. If Q is 0.6, P is 0.4. If P is 0.4, then this is going to be 0.16, and this is going to be 2PQ, which is nothing but 6 into 4.48. So I started with homozygous recessive frequency, and based on that we were able to calculate heterozygous frequency as well as homozygous dominant frequency in the population. How could we do that? Based on this calculation. That's it. Take any example and prove this, and then say, in general, Hardy-Weinberg does not, the flowchart that is showing the same, this flowchart shows the same logic. So then say, this does not hold true, because in real populations we have many mechanisms which guide evolution. And what are the mechanisms? These are mutation, genetic drift, gene flow, non-random mating, and natural selection. And explain each. Mutation can be of two types. You can have point mutation. You can also have chromosomal aberration. Genetic drift again of two types, founder effect and population bottlenecking. And if possible, give examples for each. We see this founder effect in case of this so-called Pingelap Island case study. Actually, this can be used for both. Then gene flow is migration. Non-random mating again can happen in two ways. Non-random mating can be because of consanguineous mating, consanguineous marriages, or non-random mating can also be because of assortative mating, where people who have certain traits want to marry others who also have similar traits. So we can talk about non-random mating. An example of non-random mating is the Amish people. The Amish people, in whom because of non-random mating there is a very high incidence of Ellis von Creveld syndrome. And finally, we can talk about how natural selection also creates and distributes variation. That's it. So Hardy-Weinberg equilibrium is no big deal. Just that students don't know that you have to write all these steps. You just write some steps and then conclusion. In conclusion, you say Hardy-Weinberg equation has quantified evolution. Until now, evolution was only studied in qualitative terms, but now using this hypothetical equation, we can quantify evolution. So the equation talks about a hypothetical scenario, but in real world, populations always deviate from it. Whenever they were deviating from equilibrium, then we have to assume that one of the factors is driving the evolution, and we have to explore which factor is driving to what extent, and that is how we have quantified the entire evolutionary process. That's it. Next is polygenic inheritance. Very simple. So define what is polygenic inheritance and start by saying that this is an exception to Mendel's theory, where Mendel believed that there only one gene will influence one trait, and Mendel believed that there is no. So this left column talks about Mendelian thinking. He believes one gene, one trait, no influence of environment. And but polygenic traits are those which are exception to Mendel, where multiple genes influence a trait, and the expression is also controlled by environment, not just genes. And polygenic traits follow what is called as normal distribution. In case of Mendelian traits, Mendel believed that it will be discrete. You will have few discrete categories, discrete distribution, but polygenic traits don't have discrete distribution, but they have what is called as normal distribution. What is normal distribution? It is to vary like this. So this is an example of normal distribution curve for height. So we can use this diagram and then discuss the examples. What are the examples of polygenic inheritance? Skin color. In this case, they are talking about height and so on. And lastly, significance. Polygenic inheritance helps us understand the role of environment, and we can study this inheritance using methods like twin method. That's it. So polygenic is very, very easy to understand. Oh my god, there is some earthing issue. Right, even eye color follows polygenic method. Next, Mendelian inheritance. Even here, students have a problem because they don't have framework. So how do you write an answer on Mendel? First, again, intro. Gregor Mendel, Austrian monk, gave some of the first principles for genetics. Then discuss his experiments. What experiments did he perform? Not all are needed, just he performed two main experiments. Focus on the two main experiments and then discuss his laws. Again, here not 5-6 laws, but the three main laws. Law of dominance, law of segregation, law of independent assortment, and then give examples of Mendelian inheritance. But then we know that Mendelian inheritance is not perfect. We know that there are many deviations to Mendel. So talk about the deviations and then conclude with significance. Significance, nothing but Mendel's theories are good, but they are not perfect. So Mendel's work laid the groundwork for further studies in evolution, and later on with the discovery of modern genetics, we now understand inheritance better. So just do all this. So what are these details? First, talk about his experiments. First, he crossed two true breeding plants for that vary in a single trait. Then he crossed, this is not needed, this you can skip. Then he did second experiment where he took a dihybrid cross. So show how he did. Discussion jarugutundi tarvata chuddam. So first experiment he takes true breeding plant, second experiment he takes a dihybrid cross and he crosses them. Now you need not discuss the outcome and all this because that space won't permit it. Just introduce the experiment. Then discuss his laws. What is the law of segregation? Next, what is the law of dominance? Second, what is the law of independent assortment? Then some examples of Mendelian inheritance in humans. Sickle cell disease, PKU, cystic fibrosis, all these are examples of many diseases which follow Mendelian pattern in case of humans, including things like color blindness. Color blindness also follows Mendelian inheritance. Then discuss some deviations. So Mendel's laws are good but not perfect. So there are many deviations to Mendel's laws, and here we discuss what are those deviations. What are the deviations? Polygenic inheritance, codominance, multiple alleles or incomplete dominance, pleiotropy, sex-linked inheritance, modifying and regulatory genes. So all these are deviations to Mendelian inheritance. Mendel thought these three laws are perfect, but in reality, heredity and inheritance is much more complex. And from here we can see that there are many nuances to how heredity operates. So you can have the role of environment. These two are same and these two are same actually. So polygenic traits are possible, modifying genes and regulatory genes are possible. Sometimes you can have pleiotropy where a single gene affects multiple traits. You can have incomplete dominance, you can have codominance, and you can have sex-linked inheritance. And none of this Mendel could envision. That's all. And conclude by saying that his work laid the groundwork for further development in genetics. That's all. Next, critically examine Darwin's theory of evolution. Explain how modern synthesis is very good. This is also another standard question because in evolution chapter generally they ask Darwin or synthetic theory every once in a while. So we can expect this question this time. So begin with what Darwin did. First half we will discuss briefly what is Darwin's work and Darwin's limitations. So the question structure should be somewhat like this. Darwin's ideas. Then next we discuss the limitations of Darwin. And then synthetic theory, and its ideas. And then we talk about the final significance, or finally with synthetic theory we can also talk about things like speciation and macroevolution. You can talk about speciation and macroevolution and how for this process we need something called as RIMS, that is reproductory isolating mechanisms. We will discuss these. So just begin with Darwin's theory. What are the Darwin's main ideas? He begins by saying that common descent, all species have a common origin. Next, he says that species go through change, and he defines evolution as descent with modification. And he says species multiply. Each species wants to increase its numbers. And he says natural selection chooses the one which has better traits. And he says evolution happens largely in a gradualistic pace. These are his main ideas. But his ideas were not perfect. They had many weaknesses. The major weakness is that he never understood inheritance correctly. Darwin never could explain how traits are being passed on from one generation to the other. So his major flaw lies in the fact that he proposed a not valid idea called as pangenesis to explain inheritance, and this is the fundamental issue. Along with that, Darwin also could not explain how species multiply. But how do species multiply? He could not explain this process that we today call as speciation. That is, how do new species arise from the old? How do new species arise from old? Even that Darwin could not explain. And Darwin only thought that there is what is called as gradualism, but he failed to understand there can also be something called as punctualism. So these are the limitations of Darwin. And post Darwin, we have this synthesis of ideas from different sides. One is the development that happened in genetics. Second is the developments that happened in evolutionary theory. So all these were merged by a group of population geneticists called as Huxley, Dobzhansky, Mayer and Simpson, and they created synthetic theory. So in synthetic theory there are different ideas. First point that you must write is synthetic theory redefines evolution. Evolution is no longer defined as descent with modification. Evolution is now defined in terms of concrete terms, that is, it is change in allele frequencies from one generation to the next. So he introduces the concept called as gene pool. So they are talking in terms of numbers now. They are not just discussing evolution in a vague terms. And then they also say that evolution has a two-step process. The first step is the production and redistribution of variation, and the second step is where natural selection acts on this variation. There are two steps according to them in evolution. First step is to produce and distribute variation. Second step is when natural selection acts. Darwin believed that only this is the key factor. And they now say that these are the different factors which produce and distribute variation. So the first step involves these 5 different processes, that is mutation, migration, gene flow, that is gene flow is migration, genetic drift, selection and recombination. These 5 factors contribute to production of variation, and once variation happens, natural selection acts on that variation to select the fittest. The natural selection itself can be of 3 types: directional selection, stabilizing selection and disruptive selection. This is the 3 ways in which selection can act. And then he says we have to also separate between microevolution and macroevolution. Synthetic theory says these are 2 different processes. Microevolution is what Darwin studied, that is finches with different beaks. But they say there is also something called as macroevolution. So macroevolution is the process that we call today as speciation. That is, how do new species come from old species? How did we begin with bacteria and how did we end up with a multicellular animal? That is speciation. Microevolution is nothing but how did, let's say, Homo erectus have regional variation? That is microevolution. Macroevolution is how did Homo sapiens come in the first place from, let's say, an aquatic animal? So macroevolution is about evolution in a large time scale. Micro is evolution in a limited time scale within a particular species. So they say Darwin's focus was largely this. Darwin could not explain macroevolution well, and they also go about explaining macroevolution or speciation. So this is speciation is a process by which new species emerge from a earlier species, and they give a new concept for species. They redefine species. Species is defined as interbreeding population which are reproductively isolated from other such populations. So what is a species? According to them, a species is an interbreeding populations which are reproductively isolated from other such populations, and they propose that for speciation we need something called as reproductive isolation or geographic isolation. Now these are more detailed, so I won't explain this, but if you are interested, you can go through them. I'll share the slides. So they say there are three ways in which speciation can occur. You can have allopatric speciation, you can have geographic speciation, and you can have sympatric speciation. These are the three ways in which speciation occurs, and for speciation to happen, we need RIMs or reproductive isolating mechanisms, and these RIMs can be of two types again. Some are pre-mating, others are post-mating mechanisms. Pre-mating isolation is what we generally discuss, that is habitat, they live in two different habitats, or they operate at two different times. One species is in night, other species is in day, or they have different behaviors, that is their sexual behaviors are not attractive to each other, or mechanical incompatibility, that is where the biological parts cannot fit with each other. These are pre-mating isolating mechanisms. You can have post-mating, they can have intercourse, but after having intercourse, there can be different kinds of incompatibility, like sperm and egg don't match, zygote becomes unviable, or fetus becomes unviable, or offspring is unviable, or offspring is born but is born as sterile. So these are the mechanisms. So now I don't expect you to read all of this in one go, but at least try to cover different things. So let me revise synthetic theory because we touched a lot. Under synthetic theory, we begin with defining or redefining evolution. We first redefine evolution in terms of allele frequencies, and then we call evolution as a two-step process. Step 1 is to produce variation, and this variation is produced by 5 factors. Step 2 is to distribute variation, and this is done by natural selection, and natural selection, as we discussed, is of 3 types again. And once this happens, the third step is to think in terms of micro and macro evolution, and fourth step is to discuss speciation. For speciation, we require reproductive isolative mechanisms, and these mechanisms again are of 2 types: pre-mating and post-mating. And the last idea is that evolution is of pace of what is the pace or tempo of evolution? It can be 2 things. It can be 2 ways. I think I missed the figure. It can be 2 ways. One is gradualism. Darwin thought there is only gradualism. The other can be punctuated equilibrium or punctuated evolution. So you can show it diagrammatically. Gradual evolution is something like this. Punctuated equilibrium is somewhat like this. This is punctuated equilibrium. So it shows both of this. That's it. That is Darwin. I think we discussed Darwin in great length. 4B is recombinant DNA technology. I think this is a SNT question. I won't discuss this at much length. Recombinant DNA technology is very, very simple. It is also called as genetic engineering. So just that students are not discussing some keywords here. Recombinant DNA, or it is also called as genetic engineering. So here first define. What is the definition? First, it is a technique of introduction of a manipulated genetic material into a host cell in such a way that it replicates and passes on this modified DNA to the progeny. And then discuss the tools used, talk about the different tools used. Most of you are not discussing the role of restriction enzymes. These are the most important things here. Without restriction enzyme, nothing happens. And then once you break it, you have to fuse it again. For that you use DNA ligase. And 3, you need a vector to carry the DNA that you want. And vectors are of 2 types. You have plasmids, cosmids, and so on. So these are the different tools that we use. And then discuss the steps. Then draw the diagram. Lastly, applications. Very simple and straightforward question, no big deal. In applications, you can talk different levels. What are the applications in agriculture? What are the applications in the field of medicine? In environmental cleanup, like bioremediation and so on. So you can discuss all these different applications. Next, 4C is again similar to 3A. I was forced to repeat the question in a different way because given the small number of topics, there were very few unique questions. So 4C is just like 3A, so we can skip that. Next is 5A. So we'll discuss 5A and 2E together because both are about chromosomal aberrations. So you have to begin first. If it is just Downs and Edwards syndrome, just briefly introduce what is an aberration. Both these Downs and Edwards syndrome are numerical aberrations. So just talk about numerical aberrations, that's it. And whenever you talk about numerical aberrations, you have to discuss these things. What is a numerical aberration? It can happen in 2 ways, polyploidy, aneuploidy, and then polyploidy can also happen in terms of monosomy and trisomy. So this whole thing must be discussed like this. Polyploidy having an extra set of chromosomes, aneuploidy an extra or missing chromosome, and aneuploidy can happen in terms of monosomy and trisomy. So this must be written first, and then you simply talk about how does numerical aberration happen? What is the key point in which numerical aberration happens? Non-disjunction. You should talk about non-disjunction. This is skipped. Most students do not write about non-disjunction. So talk about how we have this whole thing happens in meiosis. It can happen in meiosis 1 or in meiosis 2, and the whole issue is that non-disjunction, that is a chromosome fails to separate in the right way. That leads to numerical aberration, and after writing that, then you can talk about Down syndrome, what are its symptoms, and you are also not writing the karyotype. You have to write this 47 plus 21, that is trisomy 21, that is Down syndrome, and list out 4-5 symptoms. Edwards syndrome, likewise trisomy 18, 47 plus 18, and list out a few symptoms. That's it. That is how you can answer 2E. So when you are writing 2E, introduce what is an aberration, types of numerical aberration, source of numerical aberration is non-disjunction, and in case of Down syndrome, it is non-disjunction at 13. In case of Edward, it is 21. So talk about each of them and then symptoms. Conclusion, we can say aberrations cannot be cured, they can only be prevented. So genetic counseling should be promoted in greater scale such that people with the abnormal conditions don't come together and mate. So genetic counseling and genetic screening is a solution to reduce the burden of aberrations. That's all. Now coming to 5A, 5A is also simpler, is also similar, except that here you have to go in some more detail. So you first begin with what is an aberration, because the first part of the question is asking discuss chromosomal aberration. So what is an aberration? Here aberration can be of two types: structural, numerical. And why do they happen? They happen because of problems in cell division. Structural aberrations can be of different types. These are the different types of structural aberrations, that is deletions, duplications, translocation, inversion. In fact, you have many of them. So these are the different kinds of structural aberrations. One is deletion, second is duplication, then you have translocation, then is inversion. Most of you are writing these four, but there are three more: isochromosome, ring chromosome, and chromotriepsis. These are the different ways in which structural aberrations can occur. Right? And then again talk about numerical aberrations. Numerical aberrations can be in this way. One way to understand numerical aberration is polyploidy, aneuploidy. Another way to understand numerical aberration is whether it is sex chromosomal aberration or autosomal aberration. After writing all of that, then we say Klinefelter and Turner are numerical aberrations in the sex chromosomes. So you write this, and numerical aberrations can again be understood in two ways. One is polyploidy, aneuploidy. The other is it can happen in sex chromosome or can be in autosome. And sex chromosomal aberration is what we see in case of Klinefelter and Turner. That is what we have to write, and then we go into discussing Turner and Klinefelter separately. And even here, because they are numerical, explain in numerical what happens. If possible, if you can draw a simple version of the diagram showing non-disjunction again. How because of non-disjunction you can have monosomy, this is trisomy, and this is euploid, and again monosomy and trisomy. How a defective cell division can create defective daughter cells, and if one of them is fertilized, then you end up getting a zygote which suffers from numerical aberration. So this happens because of non-disjunction at meiosis 1, and this is because of non-disjunction at meiosis 2. This is just for information, not needed. And then you talk about Turner syndrome. What is the karyotype? 45 XO happens in how many people? 1 out of every 2500, and the basic symptoms. Klinefelter, what is the karyotype? 47 XXY, 1 out of every 500, and the basic symptoms again. And the conclusion is very similar again. You will say that these cannot be cured. The best way forward is to prevent them through genetic counseling and genetic screening. That's it. Next is various methods used in genetic study of man. So this is again 9.1. What students missed is first write intro and then discuss the need for these methods. What is the need for these methods? Why do we have the need for these indirect methods to study man and man's genetics? Because one, human reproductive cycle is very long. We take 9 months to reproduce. Second, not that just it is long, the litter size is small. You have one baby born in every 1 or 2, maximum 3 in every birth. Third, many traits traits occur in polygenic state, so we can't study individual traits in humans, very difficult. And of course, there are the serious ethical issues to manipulating humans for the purpose of genetic studies, ethical issues. Excuse me. So given these reasons, we can't study genetics in humans directly. You can't you can't grow lab rats, you can't grow lab animals in case of humans and just raise some people in lab to test them for various genetics. We can't do that. So in case of humans, because of these different reasons, we have to study them indirectly. We have to study them in their natural condition, and that is why we have evolved different methods. And here now discuss all the methods briefly. Don't leave anything. Pedigree, twin, co-twin, foster child. These three are actually very similar to each other. Then we have karyotype or chromosomal analysis. Then we have immunological methods. Then we have 7 biochemical methods. So discuss these different methods and close the discussion. That's it. Now the last question. Last question is 5C. First is, what are lethal and sublethal genes? This is again very straightforward. This is from I think 9.2. So talk about the different one. Where is this? Yes. Talk about what is lethal. Lethal genes are genes which can kill the individual, and here you have different categories: sublethal, semi-lethal, conditional lethal. So define clearly what is sublethal. This causes death before reproductive age, semi-lethal after reproductive age, conditional lethal only in some conditions, and then give examples for each of them. In case of lethal, again you can classify them into dominant, recessive. Within dominant, again you will have sublethal, semi-lethal. Within recessive, again you will have sublethal and semi-lethal. Just describe what they are with examples. And then if you want, you can also discuss some of these major lethal genes like cystic fibrosis, albinism, Tay-Sachs, thalassemia, sickle cell anemia. This I'm expecting question on this year, one way or another, because India recently launched a sickle cell anemia mission. And Huntington's, achondroplastic dwarfism, all of these are examples of different kinds of lethal genes. And as we know, lethal genes cause death to the individual, and what extent of death depends on whether it is semi-lethal or sublethal. That's it. And what is next? So given that, what can we do about it? Since it's a genetic issue, we can't have a cure. A cure is not possible. The only remedy is either you manage the disease, or the only other alternative is gene editing. And what is the most promising gene editing technology that the world has today? CRISPR-Cas9. And here discuss CRISPR-Cas9 and its principle. So what is CRISPR-Cas9 principle? It involves CRISPR and Cas9. Cas9 is the molecular scissors. So here we use a guide RNA to open the DNA helix, and this guide RNA binds with the DNA at a specific point. It's like somewhat, the diagram looks somewhat like this if you draw it. The double helix has been opened. We have genetic sequence. and it looks somewhat like this, the guide RNA. So the guide RNA opens this and you have Cas9 is this is a scissors. So this is a scissors which cuts the DNA at the place where the guide, this is a guide RNA. So guide RNA binds with a specific place where we want to edit the gene, and CRISPR, once the guide RNA has finished binding with the DNA, Cas9 then cuts the DNA at the place where the binding has happened. This is cut, this gene gets removed, and then in the third step, this is the first step where guide RNA binds. Second step is where Cas9 cuts, and third step is once the segment is cut, then the body's natural DNA rebuilding mechanism is used to then substitute for these genes. That is CRISPR-Cas9 technology. And then briefly discuss its applications. Again, applications are manifold. After describing the technology, briefly talk about applications, at least 4 applications, and then concerns. In concerns, what you must definitely talk about is the He Zhuang Kui. I think it is, what is the exact spelling? He Jiankui, yes. He Jiankui. Controversy. This is also called as Lulu Nana controversy, and then this concern over creating the so-called designer babies, and then ethical concerns, whether we are using this to edit germline cells, because if you edit germline cells, then you edit your cells, as a result even your children inherit modified cells, and the concern of using this for designer babies. That's applications, medicine, treatment. We can use this for, it is being used to create vaccines. For example, many vaccines were created during COVID time using CRISPR technology. It is used for diagnostics. It is being used in agriculture, and so on. That's it. That's all. We can use this for creating pest-resistant plant, high-yielding plants, or things like reviving endangered species, reviving extinct species. So all of this can be done using CRISPR-Cas9 technology. So that's it. So that's how you can look at this. So with that, we will close this discussion, and we'll again meet for test 7, right? Any queries, Bharat? Clear? All right.